My name is Seth Bordenstein. I'm a member of the Evolutionary Studies Initiative, and it is my great pleasure to introduce Dr. Corrie Moreau, who is the Martha N. and John C. Moser Professor of Arthropod Biosystematics and Biodiversity at Cornell University in the departments of both entomology and ecology and evolutionary biology. With one genus and 3 species named after her, she is also the director and curator of the Cornell University Insect Collection with over 7 million specimens. Dr. Moreau earned her PhD in evolutionary biology from Harvard University under professors Naomi Pierce and the late E.O. Wilson, and was a Miller Fellow at the University of California, Berkeley. And before this, she completed her undergraduate and master's degrees at San Francisco State University. Dr. Moreau was elected a Fellow of the Royal Entomological Society in 2021, a Fellow of the Entomological Society of America in 2020, an American Association for the Advancement of Science Fellow in 2018, a Kavli Fellow of the National Academy of Sciences USA in 2016, and a National Geographic Explorer in 2014, and finally highlighted as a Woman of Impact by the National Geographic Society in 2018. Dr. Moreau's elegant research on the evolution and diversification of ants and their symbiotic bacteria couples both field-based research with molecular and genomic tools in the lab to address the origin of species and how co-evolved host microbe systems benefit both partners. Also, she pursues questions on the role of biogeography, trait evolution, and symbiosis in shaping macroevolutionary processes to better understand broad-scale evolutionary patterns of life. And so in addition to Corrie's passion for scientific research, she is also engaged with efforts to promote science communication and increase diversity in the sciences. I know we all are thankful that you're here today. Welcome, Corrie.
Great, thank you so much for having me. I'm really excited to share some of the work that we've been doing in my group. So I thought I would start with what I see are sort of the big challenges and solutions in biology. One, I think that discovering and documenting biodiversity is one of the challenges we still face. But I would argue that natural history museums are actually revolutionizing science in ways that we hadn't necessarily predicted. And I would argue that we all must work to make science more diverse and inclusive. Sorry, so why is it so important to document and discover biodiversity? One of which is that we know very little about the natural world on our planet. And of course, there's lots of things that humans can benefit from knowing about the planet. But in addition, these are sort of these interwoven tapestries that are complex ecosystems where if you pull the thread of one species out, it has huge implications and impacts across ecosystems and food webs. And although most of my work focuses on running around beautiful rainforests like this, what we see the world over is that we're seeing the conversion of forests for human modified habitats. Now, this is in Uganda, Africa, but I guarantee you this is happening right outside your door. We need more and more space to grow the amount of food that we need to, sustain the human population at the level we have. We need to extract more and more resources as we advance our technologies. And so we need to start thinking about how can we ensure that we're setting aside enough of the natural world that we can maintain the ecosystem services provided by these habitats, but also begin to understand all the organisms living within them. So I am a huge fan of natural history collections. What you're looking at here is a drawer of ants that I collected, carefully curated and deposited into a natural history museum. And I think part of what makes natural history collections so exciting is all of the ways in which we can leverage these specimens. Of course, we can do the thing we've traditionally done, which is study the anatomy. So we might study things about the morphological structures, the external characters, that they're used often in taxonomy. But of course, there's all kinds of information, important information that's on each and every one of those labels associated with a specimen in a natural history collection. And what that allows us to do is to understand where species were in the past, whether they might be increasing, shifting their ranges. We can actually ask questions about when a species is discovered in a new place. Is it actually new to that habitat? And if so, where is its origin? Is it a potentially invasive species? We also might want to use sort of climatic niche modeling to try to understand, okay, well, if this is where these species are currently found, what are the implications for climate change? Where do we expect these species to increase their range to? Of course, we can actually go in and sequence genes or parts of genomes, and in some cases, whole genomes from these museum specimens. And this is important because it's not just that we can study extinct species this way, right? And there's no other mechanism to study them that way. But we can also actually look at things like genetic change through time. So even if a species is, doing quite well in the environment, we might want to understand something about, are we losing genes of interest or genetic diversity? More and more, we're beginning to understand that we can study things about the microbial communities associated with each of these individual species. And then ultimately, there's lots of ways in which we can use these collections that are sort of novel in the ways that we've thought about collections. And that has to do with nature-inspired design, and I'll talk through some examples of that. So I would argue that museums and specimens are critical for scientific research across fields, and they're foundational for many of these fields in things like describing population species and the tremendous diversity of life on the planet, for improving public health, agricultural practices, and even food security. We can actually monitor environmental contamination. So people have gone in and sampled things like heavy metals on the outside of organisms and seen whether they change through time, if there's a critical period in which we start to see an increase in environmental contaminants. We can ask questions about might be sort of these vectors for harboring diseases that may emerge, like COVID has done. We can work on conservation of endangered and threatened species by using these collections. And of course, we can use them to understand these bio-inspired designs across disciplines. And so, for example, we actually had a material scientist partner with the collections manager at the Cornell University Insect Collection, because he wanted to understand why, when you see a raindrop fall and hit an insect, why is the insect not blown to pieces, right? And that has implications for how they make materials. So they spent lots of time dropping lots of water droplets off of insect wings and watching how the wings structured, fractionated the water so that it wasn't actually harmful to the insect. And of course, we can do things like study biological invasions and the impacts of global climate change. And in many cases, we can never really understand how climate change is impacting species diversity without going to natural history collections. I'd also argue that we all must work to make science more diverse and inclusive. We know that across all of the STEM fields, so science, technology, engineering, and mathematics, there's not currently enough trained candidates. We know that when we have diverse teams, we have increased innovation, increased productivity and problem solving, increased teamwork, and we're more likely to attract and retain talent. And you don't have to believe me. These are two studies that have demonstrated that when you have diverse groups, your science is much more impactful. And I would argue that museums and diversity are not mutually exclusive. You can actually work together to sort of forward both of these missions. I published a paper a few years ago with some colleagues arguing that the digitization of museum collections has the potential to sort of make these accessible collections around the world and increase the reach that our science has to communities that may not always have access to resources like in these natural history museums. So, but scientifically, the things that get me really excited are trying to understand the processes that have generated the biological diversity we see around us. When I step out into the rainforest or in my backyard, what I'm often thinking about is, what are those abiotic factors? So things like temperature, rainfall, precipitation, soil pH, that are dictating what species we see there. But I also think a lot about the biotic interactions. How are the predator-prey dynamics playing out? And what about all these forms of symbiosis? And so ultimately what I'm trying to understand is, how does symbiosis have an effect both at the short-term ecological scale, and then how does that play out on the longer-term evolutionary scale? And so as an evolutionary biologist, one of the things I get most excited about is thinking about the world through the lens of a phylogenetic tree. And the thing that's great about a phylogenetic tree is it doesn't just tell us what species are related to who, but we can actually start to do things like map the evolution of traits. Has something evolved once and then led to prolific diversity within that lineage? Has it evolved independently multiple times? And if so, why? Is there some sort of ecological pressure that's sort of leading to the evolution of these novel traits? We can ask questions about, when did a lineage split into multiple lineages? When was something very widespread across a large range and then a mountain arose and divided the populations? A lot of the work I do in my lab is very molecular based. And so we might want to ask questions about rates of change along lineages or branches or clades. And so why is it some parts of the tree of life have faster rates of molecular evolution while others have slower? And if we have some kind of external information, we can put a time frame on that and try to understand, when did those traits arise? When did that speciation event happen? So as Seth mentioned, one of my PhD advisors was E.O. Wilson, and he once said that ants are the little things that run the world. And of course, as an ant biologist, I 100% believe that. But part of the thing that makes me so excited about working with ants is that they engage in symbiosis across the tree of life. So we know they engage in symbiosis with other animals. So this is a scale insect here that's produced this little droplet of honeydew. And in return for receiving this little sugary snack, the ant will protect that scale insect from predators, parasitoids, and anything trying to attack it. We know that they engage in symbiosis with many kinds of plants, both in sort of obligate interactions like this ant-plant mutualism here, which we've done some work on, I won't present on today, but in many cases in very facultative ways, which benefit the plant nonetheless. We know that they engage in interactions with lots of kinds of fungi. And of course, the sort of classic example of that is the fungus growing ants. But not all of these symbiotic interactions are positive. And so this is actually a fungus, the zombie fungus, that manipulates the behavior of the ant to maximize the reproductive output of the manipulative fungi. And I've done some work on that group of ants with David Hughes, who's done some magnificent work on these fungi. And more we're beginning to understand about the microbial world, the bacteria, the fungi, and even all kinds of other invertebrates that are invading their hosts. But as I've started to think about all the things that are interacting inside of the organism, that the ant itself is a walking rainforest full of life, I now think that they're the little things that run the little things that run the world. So if you sort of think about plant diversity through time, one of the things we know is that there was a lot of sort of diversity that happened in the past, but really now we're in this age of angiosperms and they've sort of taken over global dominance. So what I'll do is I'll walk you through some work in my lab that we're doing looking at the evolution of ants themselves at the macroevolutionary scale. Then we'll sort of look at the role of the bacteria inside of these hosts. And interestingly, as those flowering plant forests went through these bursts in diversification, one of the things that we saw is that lots of other organisms also went through bursts in diversification. So many insects that either feed directly on plants or pollinate plants went through bursts in diversification. But even some things you wouldn't necessarily predict, like epiphytes and ferns, went through bursts in diversification. And it's thought that the dense canopy created this low light environment under which they thrive. So you might not be surprised to know that ants also went through a burst in diversification correlated with the rise of the flowering plant forests. And interestingly, we also know that they expanded their diet into many plant-based resources. So they feed directly on things like these extrafoil nectaries, these little like sugar water fountains that the plant produces specifically to attract ants. We know that some ants have produced these specific food bodies so that the ants can then feed them to their larvae. We know that some plants actually have these coatings on their seeds called an elaiosome, and that's to attract the ant to drag the seed away from the parental plant, so the parental plant is not in direct competition for resources with its offspring. And then many ants feed indirectly on plants through these sap-sucking insects, which are mediating that interaction. So as the sap-sucking insects feed on the plant, they produce that honeydew droplet, and many ants feed almost exclusively on those honeydew droplets. So what we want to try to understand was, is there some sort of link between the evolution of plants and ants? And how did these associations rise? So we have all these specialized structures in plants, and does it correlate with where we see, changes in the behavior of ants? So is there some kind of evolutionary sequence by which ants became reliant on plants? Inferred a very large plant phylogeny. This was actually one that had already been published. We just added a few ferns at the base of the tree because we wanted to account for the fact that some ferns also produce extrafloral nectaries. And then we went in and did ancestral state reconstruction on these ant-associated traits. So we looked at how many times did extrafloral nectaries arrive, and elaiosomes and domatia. And since this was a time-calibrated tree, we could actually ask, when did those things happen? We next inferred a very large species level ant phylogeny with almost 1,700 species of ants included. And we again did ancestral state reconstruction, but on things like diet and where these ants are foraging and building their homes. And this is work that was really spearheaded by a former postdoc in my lab, Matt Nelson. So before I walk you through what we found, I just want to remind you that sort of the earliest ants, their ground plan was that they nested in the ground, they foraged on the ground, and they were predatory. They were eating other animals. And if we sort of look at what we found as we reconstructed the evolution of these interactions through time, the first thing that we see is that ants begin to forage arborally. So they're no longer staying exclusively on the ground to look for food resources. They're going up these trees to try to find prey. Next, we see that they begin to include plants in their diet. And this is likely through things like licking wounds, but also maybe even stealing nectar. Next, we see that they don't just sort of go up into the canopy to look for food. They actually begin to nest arborally. So they're taking advantage of the fact that there's these cracks in the trees and these hollow twigs in which they can live. And so in this case, they may, in some species, not even going back down to the ground at all. We see the plants respond and have the evolution of these elaiosomes and eventually the domatia. And so what this shows us is that ants utilize plants long before plants had evolved these specialized structures, which is exactly what you would predict. That we see this incremental sequence of evolution that led to this reliance of ants on plants and vice versa. And so we have ants that go from being predatory, nesting on the ground and foraging on the ground, to then being able to forage arborally. They begin to include plants in their diet, and eventually they nest arborally as well. And we see the plants respond where we first see the evolution of these extrafluonectaries after we have ants not only foraging arborally, but beginning to include plants in their diet. We see that the plants are also responding by the evolution of elaiosomes, so taking advantage of this army of ants walking around on the ground to do their seed dispersal. And eventually we see the evolution of domatia, these specialized structures just to house ants to live permanently within the trees. And although we hadn't set out to test this and it had been speculated on in the ant literature, one of the things we were able to demonstrate is that for an ant to transition to being entirely herbivorous, and this has happened independently multiple times across the ant tree of life, we always see that they transition through an omnivorous stage from the predatory stage. So we never see a jump from predatory to herbivorous. In addition, once a species becomes or lineage becomes herbivorous, we rarely see reversions to omnivory and we never see reversions to predation, suggesting that once you become sort of locked into this highly specialized diet, it's quite difficult to revert. So hopefully now I've convinced you that ants have quite a long and intricate history with plants. And that, since we know that herbivories evolved multiple times across the plants, it begs the question, could they be using symbiotic bacteria to sort of facilitate this novel shift? So I would argue ants are a great group to study gut microbes for several reasons. One of which is that we can study things like diet and nutrition. So we can ask the question, so do we see the acquisition of different or even similar groups of bacteria every time we see the shift onto an herbivorous diet or a novel diet? We can also ask questions like, what happens when we manipulate the diet of these ants in real time and watch what happens to their gut communities? We can ask questions about transmission. So since ants are social organisms, we can ask questions sort of at an evolutionary scale, like how are they passing, these microbes that might be necessary for their survival. But even within a colony, how are pathogens spread around? How is the system stratified to protect the most sensitive parts of the colony? When we do have these bacteria that are associating with their host for long evolutionary periods of time, when should we expect or predict to see sort of host and symbiont sort of co-diversifying or having a shared evolutionary history versus not? And then since microbes are everywhere and ants are almost everywhere except Antarctica, we can ask the question, are they just acquiring these microbes from their local environment? So when I first started doing this work with my longtime collaborator, Jake Russell, I sort of joked that I felt like we were these rainforest explorers, right? So we didn't have any idea really much about the bacterial diversity associated with ant guts. And so the first thing we had to do is just go out and find it, gather every weird thing we saw. So what we did is we first sampled almost 400 individual ant guts from 150 ant genera and then sequenced the bacteria associated with them. And not surprising, we found a lot. But what really jumped out to us is we saw an enrichment of this group of bacteria, the Rhizobiales. Now, the reason that we were interested in this to find a group of bacteria in the same group, not highly related, but there is a group of bacteria called the Rhizobia, which we know fix atmospheric nitrogen for their plant hosts. So in return, the plant provides these little nodules and a carbon source for the bacteria. And then the bacteria actually have the mechanism within their genome to fix nitrogen, and then they provide it to these leguminous plants. So could we see some similar roles for the upregulation of the host's nutrition through the use of these bacteria? And another hint that suggested that it might be important based on diet is this. So what you're looking at here is the trophic scale. So it's sort of the food web from things that are highly predatory, like army ants out here, all the way through things that are very herbivorous or feeding very low on the trophic scale. So what you're looking at is the ratio of heavy to light nitrogen as it moves up the food chain, of course, grounded in known herbivores and known predators and the plant base itself. Every one of these orange diamonds is an individual ant genus with multiple individuals sampled for their stable isotopic signature. And that's plotted along the y-axis. And on the x-axis, and then on the y-axis, we have whether there's an association with Rhizobiales, that group of bacteria that we saw enriched across ants. And interestingly, we only say an association with Rhizobiales for ants that are feeding lower on the trophic scale. But if you have a keen eye, you might have noticed that there are 2 points that are quite low on the trophic scale and have no association with Rhizobiales. And those were the carpenter ants and their relatives. And that's one of the few groups of ants that we actually knew anything about their bacterial communities. And within these carpenter ants, they have a long association with a group of obligate bacteria that are associated with a bacteriocyte, a specialized structure within the ant's body, specifically to hold these bacteria. And we know that they upregulate the host's nutrition. So it appears if you're going to feed low on the trophic scale, you need some group of bacteria associated with your gut. What you're looking at here is a phylogeny of the bacteria, but color-coded and named by the host or environment from which it was found. And what you'll notice is that almost all the bacteria that came from ants all fall in this one specific Rhizobiales clade. And all of these ants within this clade are mostly known to be herbivorous. The couple examples we have elsewhere in the phylogeny, those ants are actually predatory. So we can't rule out that they didn't eat something, and likely not something from an ant, but from a distantly related group of bacteria. And interestingly, even within that Rhizobiales specific clade, you'll notice that the names are the host names, but the sequences are the bacteria. The ants that are more similar to each other have bacteria that are more similar to them. But if we ask the question from the ant side, we actually see 5 independent associations of this group of bacteria across the ants. And it's highly associated with the ants being arboreal and herbivorous. So if these bacteria are in fact enriching the diet of the host, we'd expect to see some amount of tissue specificity. And so what we did is we went in and dissected out individual compartments of the digestive tract to look at the bacterial communities associated with them. So just to remind you a little bit about ant physiology. So this is the crop, and the crop is the social stomach, right? So this is where when an ant worker is out foraging in the environment, finds a liquid food source, they can suck in a whole bunch of it, walk back to the nest and regurgitate it to other members of their nest. And no digestion occurs there, right? It's just a holding chamber. Next, we have the midgut, and the midgut is where we know digestion begins. And then we have the hindgut, which is made up of both the large intestine, the ileum, and the rectum, which is where the waste is excreted from. So if we expect that there's some amount of nutritional upregulation, we'd expect there to see some amount of structure in the bacterial communities in these different compartments. So what you'll notice here is the crop, which is the social stomach. So each one of these bars is an individual sample. So it's a crop from an individual ant, and they're actually across many species of turtle ants. And what we see is that within the crop, although there's lots of variation across, within a single sample, most of them host only a single bacterial order. Now, when we go into the midgut, we can see that regardless of the host species, so this is across many species of hosts, the midgut is almost exclusively dominated by apoptotales. Now, once we move into the ileum and the rectum, we begin to see that the bacterial communities are pretty consistent but diverse. And then here we're using the gastro. Actually, we included the entire abdomen for some individuals because those careful dissections are quite time consuming and tedious. We wanted to know if we could just throw in the whole abdomen and would it recapitulate what we see, which it does, which is a reassuring thing that sometimes we don't have to do the dissections. But it's not just the identity of the bacteria that seems to be correlated with the digestive compartment. If we use qPCR and look at the quantity of bacteria within these guts, either pooled or looking across all the individual samples, we can see the crop that although it looked diverse across it, within each sample, although there was usually only a single bacterial identified bacterium, we see that there's almost no bacteria there, right? So this is just sort of transient, environmentally acquired contaminants. Once we move into the midgut, again, we see that there's a lot more bacteria present within the midgut. And again, we're still seeing that retained within the ileum, and then within the rectum where we know that it's excreted, we still see very low quantities of bacteria. So we wanted to ask the question, well, back in the leguminous plants, we knew that what the bacteria were doing was fixing atmospheric nitrogen. So could they be doing something similar in their hosts? We took a two-pronged approach. One, we went in and tried to sequence those genes that actually are responsible for bacteria being able to fix atmospheric nitrogen. And although we could find pieces of it, we couldn't find fully functional genes, but it doesn't mean that they're not there. But we also went in and did these acetylene reduction assays to test whether we could get the bacteria within the guts of these ants to fix atmospheric nitrogen. We've done this now across many different species, both individuals and pools of workers from the same colony. And in all cases, we've seen no signature of the fixation of atmospheric nitrogen. So if these bacteria are upregulating their host, this is not the mechanism. We have another line of indirect evidence that suggests that these bacteria are in fact enriching the host's diet. And so this is a fluorescent in situ hybridization image, a fish image taken by a colleague of mine, Peter Lukasik. And again, this is the sort of digestive compartment. The crop is out of this shot here. This is the midgut. This is that ileum or large intestine. And this is the rectum. It's ruptured on the end, but you can see the rectal pads. And these squiggly lines are called the Malpighian tubules. So the Malpighian tubules in insects are essentially the kidneys. So what these Malpighian tubules are just loose within the body cavity, and they're grabbing any of the waste products, uric acid waste, and depositing it into the digestive tract so it can be excreted. And so interestingly, where we see the probes glowing the most is right at the juncture where all those Malpighian tubules are dumping that uric acid waste into the digestive cavity. And the reason that that's interesting is that for many bacteria, they can take this, product like uric acid or urea, and then use it as the first precursor or building block to synthesize amino acids. We do have a couple of lines of direct evidence. The first comes from both genomic sequencing of the bacteria we've been able to culture, but also metagenomic sequencing. We've now looked across 18 different Cephalotes metagenomes from 17 genomes and metagenomes from 17 species of turtle ants. So it's across their about 46 million years of evolution. And we've been able to demonstrate that the bacteria within the gut retain all the genes to synthesize amino acids, suggesting that they have the capacity. But we've gone one step further. We've actually done some manipulative work, and this has really been spearheaded by Yi Hu and my colleague Jake Russell, where we went in and we took colonies of these ants, split them in two, and used antibiotics to cure or knock out or knock down their bacterial communities, left some intact. We then had some treatment groups in which we fed them this labeled urea with bacterial communities removed. They didn't give them the enriched form and left their bacterial communities intact. And we can see that the amount of these essential amino acids is more or less similar within these ants. Now, we're not testing this within the gut, right? Because these bacteria could be producing these amino acids for their own use. So although the bacteria are contained within the gut, we actually sampled the hemolymph or blood of the ant to see if we could find the signature of these labeled isotopes. And when we leave the bacterial communities intact and give them the labeled isotopes, we can see a sharp increase in the amount of these essential amino acids that are accessible to the host. And so what this demonstrates is that at least within the turtle ant system, the gut bacteria are providing these amino acids in the form of nitrogen recycling. So they're using the host's own waste and potentially some items that are being eaten in the diet, and then using that as the sort of precursor to synthesize the amino acids. We do have the situation in which these bacteria are producing these amino acids. What is the host using them for? So we explored the question of whether they could be contributing to the formation of the cuticle or the exoskeleton in these ants. And so just to remind you, in insects, the exoskeleton is essentially its armor. It's what protects it from the outside world, both from physical harm, but also desiccation and from pathogens and pests. And we know that there's a lot of variation across the phylogeny of ants, and they're sort of the physical aspects of the cuticle and the cuticle toughness. And the reason that we actually decided to ask this question was, as I've sat at a microscope dissecting out thousands of ant guts, one of the things I noticed is that the ants that had the toughest cuticles to break through were either predatory or were herbivorous, and we knew they had gut-associated microbes. So this suggests that when you have a lot of nitrogen in your diet, you may have the capacity to invest it in your cuticle. But we wanted to test, were in fact these bacteria responsible for these tougher cuticles? So we've also done some work with material scientists, and what we're looking at here is sort of the toughness or measured on this axis. And what we're seeing is there's actually variation across groups of ants, and it's not surprising based on what we know about them. And interestingly, this is to some extent correlated with cuticle thickness, which is maybe not surprising, but that's not that strong of a signature. But interestingly, if we look at the sort of physical components of the cuticle, so if the cuticle itself has all these ridges, we can see that there's a correlation with the number of these peaks and the increase in the physical strength of the cuticle. So the cuticle itself is actually primarily made of chitin, but within that chitin, there are embedded proteins. And in order for the chitin to attach to a protein, it requires a linker. And although the protein can embed inside of the cuticle, it actually requires the imidazole arm of histidine in order to join to the linker. Now, within this matrix, there can be other embedded particles, things like melanin and metals. And I won't talk about it today, but we've done some work to demonstrate that we see an enrichment of metals along the blade of the cuticle in most species of ants and in the tarsal claws that they walk on to reduce the abrasion, and that it's not evenly distributed across the cuticle. So this is some work that was published in 2015, and it's a really nice example of sort of the tanning process that leads to this tough cuticle through development. So what you're looking at here is the mandible of one grasshopper, and then this is a composite image. So they've showed it from all the way from the untanned material through the tanning process till it becomes fully tanned. And what they were able to demonstrate is that we have, during this tanning process, we have the amount of oxygen staying stable, but we see that nitrogen increases, displacing the carbon. And in addition, what we see is a sharp increase in the number of proteins and sclerotized tissue, while the amount of chitin stays the same. And that's really what leads to that toughness or strength or hardness. So this begs the question, could the bacteria in the guts be contributing to the formation of the cuticle? There has been some previous research that suggests that this in fact is true. At the time our paper was studied, there were actually 5 papers that had been published. All of them had looked at beetles, but in those studies, what they had demonstrated is that when you use antibiotics to knock down or out the bacterial communities, you see a sharp decrease in the pigmentation or melanization of these beetles, right? So we see them go from very darker colors to paler colors. We also see that there's a sharp decrease in the amount of cuticle thickness. So this is with their bacterial communities intact and then with them missing in 2 different studies. And one of the studies identified a putative biosynthetic pathway predicted from the genome of the bacterium and demonstrated that all of the precursor steps were executed by the symbiont bacteria. And the last step was actually the insect itself that finished this biosynthetic pathway. So what we wanted to know is, are there other particular components that are impacted? So what about the protein, the linker, the chitin, the melanin? And they've predicted one pathway, but is this sort of the known pathway across all insects, or would we expect to see novel ways in which this happens? And then ultimately, we want to understand whether these mechanisms are conserved across many species. So what we did is we set out to actually test this question, took colonies of this turtle ant again, split them in 2, gave some antibiotics and some not. We gave them a diet that was enriched in urea, and we ran this experiment for multiple months. And that's because When we started the experiment, in order for the larva, which is what we really want to see, because they're going to go through the diet manipulation experiment, to mature, they actually have to have adult workers within the colony to feed them. So once we had done that and labeled and enriched their diet with nitrogen 15, ran the experiments, then we pulled out the new emerged workers and actually looked at their guts. So we used liquid state NMR to explore components of the gut chemistry. We then used solid state NMR exclusively on the cuticle. So we scraped out all of the insides, dried out the cuticle, and only examined the cuticle. And in both stages, both of these cases, we're using NMR. And for those of you who maybe don't remember this, it's just an analytical chemistry technique that allows you to trace a single isotope in a system. And then we also use scanning electron microscopy to look at physical characteristics of the cuticle itself. Now, in that liquid state NMR, what we did is essentially, this is a control. We were making sure that we would see the same pattern we had seen in our previous study, which was when we use antibiotics to treat the symbiotic communities, we see a sharp decrease in the amount of tyrosine and phenylalanine that are present within the insect. So we knew that we were manipulating our ants in the same mechanism. But when we use the solid state NMR to look at the cuticle, when we compare both those that had no treatment, so the symbionts are intact, versus those that have had any antibiotics included, we can see there's a huge enrichment of nitrogen within the cuticle. So when the bacterial communities are present, we can see huge peaks. Now, if we look at the identity of all these peaks, one of the things that we see is that the bacteria that are contributing to the chitin itself and the embedded proteins. We can see it's involved in the guanine arm of histidine, the imidazole arm of histidine, as well as many of the linkers. And so this really told us that the nitrogen produced by the bacteria are in fact being used to build the cuticle. Now, coming back to the thickness of the cuticle, so these are not to scale, but you have to read the measuring bars. We can see that when we left the bacterial communities intact, the cuticle is twice as thick as when we knocked out the communities. But unlike in those beetle studies, we actually saw no impact on melanization. Those individuals were just as black as the individuals who'd been getting antibiotics. And we actually knew that. We've been doing these bacterial manipulation with antibiotic experiments for, oh gosh, I hate to admit it, more than a decade, and we never see depigmentation within our ants. So what this essentially allowed us to do is to reconstruct the biosynthetic pathway and where the bacteria were contributing in this process, and demonstrated that the bacteria contributed to the protein, the linker, and the chitin within the cuticle, but not the melanization pathway. And so I thought I'd take a moment and talk about broader impacts and outreach and science communication and why it's so important. Of course, we want to be able to communicate our research with audiences of all backgrounds. And it's not just that out of pure, unadulterated wanting to talk about what we do all the time. We actually know that an informed public is much more likely to support future efforts and make informed decisions. So science literacy is something that's critical for all members of our society. I mean, we're seeing that play out even now with things like vaccine hesitation and questions about what are the best strategies to curb this global pandemic. So we've done outreach in many different ways, and I actually personally have decided that for myself, taking diverse perspectives to be able to share my work has been effective for me. But I know a lot of people who do things like have a really popular YouTube channel and only do that. I think that whatever your bandwidth or your interests are should be what drives your science communication. So I had a long-term project in the Florida Keys trying to understand both the diversity of the ants themselves, but also our microbes shared among interacting ants within the Keys. And so while I was down there, I did a lot of collecting and interacting with local groups. And so I created this diversity poster. And this poster shared not only things about, just the diversity itself, but about the importance of ants in the environment and a bit about their ecology. I partnered with some educators to create a flyer that I went with this poster so that teachers could confidently use it in the classroom. And that poster itself actually conforms to the next generation science standards and allows teachers to be able to map it to the particular grade structure and activity that's being used within their classroom. Another thing I've done is I've been able to participate on some online videos and podcasts. And those are great because they can be as formal or informal as you like. And they can be a great way to reach audiences around the world. I sort of included English speaking here because those are usually the audiences, the language that they're recorded, at least for my group. But in this case, I worked with Emily Grassley of the Brain Scoop, and many of her videos have been translated into 20 plus languages. And so you have the ability to reach audiences you may not typically have been able to. Another thing we've done is we've actually turned over, I mean, we used to actually give camcorders now, or, some kind of recording device. Now we essentially just have students use their phones, but we actually, as part of an internship program, have them create their own videos about their experience, both doing research and what their research findings are. And it's been a really fun way for them to engage with the research. I've also worked with museums to create exhibits reaching these what we call K through gray audiences, right? So through informal learning. This is an example of an exhibit that I participated in called The Romance of Ants, and essentially took you through my pathway from me being a little girl interested in bugs all the way through my scientific career. But woven throughout that, we of course had these large images of ants and a lot about the biology of them. We had a larger than life ant farm and videos and all kinds of ways for them to interact with the materials. And in the 2.5 years that exhibit was open, we had over 500,000 visitors come to that exhibit. And I'm actually getting ready to open a public museum exhibit here in Ithaca called Six-Legged Science. It's going to be opening at the end of March. So if you happen to be out this way, we would love to have you come and see the exhibit. It'll be open through December. And this is a great mechanism to sort of share the diversity and importance of insects with really wide-ranging audiences. So what I hope I've been able to convince you is that on the research approach, using this really integrative approach to study questions about symbiosis across many timescales allows us to sort of understand things about documenting and discovering biodiversity. And hopefully I've been able to convince you that natural history museums are not these dusty old institutions with very, very little use, and that now they're becoming much more vibrant research hubs as all of these new technologies are appearing that we can leverage these collections. I would argue that all of us need to work to make science more diverse and inclusive, and that using these diverse tools can lead to questions that maybe you wouldn't always have when you only work with one sort of organism or set of questions. And I would argue that science communication can come in many forms, and it's critical to engage diverse audiences in this lifelong learning process. And so with that, of course, I want to thank the collaborators I've been able to do this research with. I really want to thank the members of my lab. I get to interact with an amazingly intelligent and fun group of people every day, my funding sources, and all of you. And if there's time for questions, I'd be happy to take some.
Thank you, Corrie. We absolutely have time for questions. As is typical for our community, we normally open the floor up to students and trainees first, and we'll use the chat box or the question and answer box for that. So I'll give everybody a second to collect their thoughts and think about what they might want to ask.
Sure, people should feel free. I know that you guys have a wide range of career stages on these calls, and so people should feel free to use it as an AMA. Ask me anything. I'm happy to share things about my career path or my passions outside of what I presented today.
So we do have one question that came in. This one's from Antonis. Excellent talk with an exclamation mark. For the macroevolutionary part of your talk, can you comment on how common convergent evolution is, either on the ants or on the plantas?
Yeah, that is a great question. There is tons of convergent evolution. Interestingly, those really seemingly complex structures like extrafloral nectaries on these plants, these little sugar water fountains, they have evolved independently thousands of times across plants. And it seems that their only function is to attract defensive arthropods, mostly ants, sometimes mites. And it's not a one-to-one relationship. So it's not like if you have an extrafloral nectary, only the ants that have a huge venomous sting and are very aggressive come drink from your water fountain. The ones that are chickens and weak also will. But there's been some sort of exclusionary experiments that have been able to demonstrate that just the presence of any ant, whether it will defend the plant or not, is enough of a deterrent for herbivores to say, I'll just try to find a plant that doesn't have ants. So just the presence of ants on plants, it actually can be a deterrent for herbivory. So we actually see it on both the ant side and the plant side, lots of convergent evolution.
Corey, on the side of these symbionts that appear to be long-term associated with ants and maybe co-diversify with ants, have you been able to look at museum specimens and go backwards to resurrect some of those strains and look at their genetic divergence?
Yeah, unfortunately not, at least not for ones that are put on pins. We've tried, it doesn't, I mean, what happens is that because during the drying process, really you have a lot of rot happening on the inside, right? Since we don't preserve them in any way. But there is hope for specimens that were preserved in ethanol or in now cryogenics is much more common. But even then in a lot of museum collections, there was this trade-off between the highest percentage of ethanol that would decrease the chance of growth of, fungus and things that are unwanted, and a high cost of ethanol. So most museum collections are preserved in 70% ethanol, which is not ideal to reconstruct these microbes, but it may make it more feasible, especially now that we have more and more of these sequencing technologies that can leverage this chopped up or broken DNA. But I do know that for many other groups of organisms, like many vertebrates, when they actually skin them, they actually pull their guts out, put them straight in ethanol or in the the cryogenics, and then they might dry the skin. So for, maybe it doesn't work in ants or insects, but there are lots of groups in which now people are going back and doing exactly what you suggest, Seth.
Neat. And then I had a second question, which is for the chitin part of this. So I guess, do you look at this as sort of the host is evolving to sequester symbionts for building up their chitin, an exoskeleton? And if you don't, then how else do you look at it? And then on the flip side, what is there anything in it for the symbionts to do that and gain a benefit? Yeah.
So interestingly, one of the things we know is that when insects have, I don't say an individual insect, but when we look across diets, so when insects have a huge amount of nitrogen available in their diets, which usually means they're predatory, they often have harder exoskeletons. We're now seeing that in many species that have gut symbionts that can provide that nitrogen to them, we also see an investment in the cuticle, right? The exoskeleton. So it suggests that you would always want a thick cuticle if you could have one. We know that it's really important for desiccation resistance, for keeping out pathogens. It protects you from the outside environment, but it's costly to make. And so it's a trade-off of when you invest in it. And whether it's actually beneficial to the microbes, that's actually interesting, because I'm going to tell you a roundabout why that I think it's true. Most of these herbivorous species are living in the canopy. The canopy is one of the most dry and hot places in a stratified environment for most ants. Things that live in the ground, they can sort of buffer themselves from changes in temperature very effectively. And then if you're sort of living in the understory, it's not as hot. And most of the ants now that we know are entirely herbivorous and rely on gut microbes to persist are the ones living in that canopy, which, we know the thicker your cuticle, the less likely you are to desiccate. And so maybe it's an inadvertent protection for the bacteria as well.
I love that. That's awesome. How do you test that? I don't know, but I'll leave that to you. Let's see, Andy has a question. So thanks for the great talk. What do you think has been your most effective or most fun outreach program?
Oh, that's a good question. I don't know if I would say, I don't know if I know the answer to that, but I will share that, I've been involved in making quite a few videos. And I remember that I made this one that I was particularly proud of. I loved the way that I communicated the science. I felt that I used the right language. I thought there was some nice, I didn't produce it, right? But there was some nice soothing music in the background. And that video, even to this day, has gotten like 1,000 viewers. And I literally think 500 of them are my dad, who just hits refresh every now and then. But then, I'll create a video that I'm just like, I partner with people who know how to do this well, and I'll be like, oh, let's just throw together this video on like bullet ant venom, and it gets 2 million views, right? So I have no ability to predict what will sort of, catch on with the public. And so I think that's why, for me, partnering with people who have a podcast audience already or have a YouTube channel already is a really smart way to get our work out there. But I hope that museum exhibits are probably the most effective. I think because you get such a diverse group of people coming through across almost all ages, and And so, you hope we have less of a measure, right? We can measure the number of people that have come through. And we've done things like spot surveys where we ask people questions pre and post, but you're not doing that every day, right? So I don't know. I'm sort of thinking that the museum, I just know what an impact museum exhibits have on me. So maybe I'm projecting.
I like this idea of put it out there because somebody's going to be listening, whether it's your dad or a million viewers. And you never know what's going to hit. And I like the idea of just put it out there. And Tonis has a question. Can you speak on how a junior scientist can establish these connections with folks that do post podcasts and videos? What kind of network are you leveraging?
Yeah, that's a really interesting question. So one of the things I often find is that junior scientists want to jump straight to the science communication, right? They want to be on a podcast, they want to be on a video, but they forget they have to have the work that they're sharing first. I mean, you can go straight into science communication and that's a whole field and you don't even have to be a scientist or trained in science. But if you want to be a scientist who's communicating your work, you have to actually produce some science that you can then share out, and you have to practice a lot. Actually, the reason I got committed and dedicated to science communication is when I was in graduate school, I had a really big paper coming out. It was right before the holidays. And so I had gone home to visit my family, and my parents didn't go to college. And so I had gone out to the bar that my dad goes to with all his golf buddies, every time since I was a kid, right? I know all these guys. They're like, hey, your dad shared you have a big paper coming out. What's it on? And I started speaking and I couldn't use non-jargony words. And I could see all their eyes glazing over, and they were so nice, they just kept smiling. And I couldn't rescue it. And I promised myself from that moment forward, I would work extremely hard to be able to communicate what I do with, diverse audiences. And so it's not easy. And as far as like finding mechanisms to connect with people, if there's a podcast you love, or, somebody who's producing videos you love, reach out to them. It's not like they usually have like, oh, I've got everything lined up for the next 3 years. They're like, oh, you want to come on the show? Great. I don't have to hunt for people to beg and plead to be on shows. And so, you may not start with, like the biggest show on the animal planet, right? You might start with something more local, but I'm sure people would be interested to have scientists come on and explain their work.
Yeah, and maybe folks can reach out to you directly and kind of get a better sense of your network. Cool. So we have a question from an undergraduate in my laboratory. This is Maddie. And Maddie asks, thank you for the presentation. We are very used to talks about coevolution of mammalian species such as humans and domestic dogs. Since to us, it is obvious that a dog wouldn't be a dog without its role in human society. How do we prove that a microbe species is necessary for evolution of its host? And is this a similar philosophical approach?
Oh, that's a tough one. First off, Maddie, awesome question. I don't think I could have asked such a sophisticated question as an undergrad. Secondly, I'll say, are we in charge of the domestication of dogs? Some people speculated dogs decided they needed us and just wouldn't leave us, and then they domesticated us. And I say that, in jest to some extent, but also I actually wonder the same thing about microbes. We often think of the larger organism as being in control, but I suspect in lots of cases, it's the smaller organisms that are controlling the system. I saw this kind of like a New York Times style cartoon early on when people started thinking about microbiomes. And it had somebody sitting down at a table in a restaurant getting ready to order their food with the menu. And the waiter walked up and said, what can I get you? And the person sitting there said, I'll have a salad. And then from their gut, their microbe said, we'll have the cheeseburger. And I have to wonder, who's actually in control? They're releasing all kinds of metabolites in our body, and maybe they're manipulating us. And another reason I think this is that there was a really elegant experiment by a guy named Todd Palmer, who's at the University of Florida. And he works on these African ant plant mutualisms. So those big acacia trees that the ants live in and defend. And there's all kinds of herbivores on the African savanna, unlike in the New World tropics, right? And he had this great idea, let's get to the bottom of this. What sort of, is maintaining this symbiotic relationship? So he just built fences around trees and waited 10 years. Now you have to have, a full professor position to be able to do something like wait 10 years, but he built these fences and waited. And what was so striking is that the trees, after some period of time, there's no herbivores attacking them anymore. They stopped producing the extrafloral nectaries and the domatia. They were shutting down the investment in the mutualism because now the mutualism was the costly thing. Why keep these ants around if there's no herbivore pressure, right? So, and this isn't across evolutionary time. This is a single plant has the plasticity to decide whether it's going to invest in this mutualism or not. And so again, is it the plant that's in control, or is it the smaller organism? And so I don't know that we have the mechanism yet to test in all these cases who's ultimately in control, but that's a great idea.
Thank you for this lovely answer. That's really great perspective to think about. Do you have any, Reese Martinez, do you have any resources you'd suggest for de-jargoning a talk? I find
yes.
I don't even know what is a jargon anymore. So this speaks to probably the comment you were making about you talking to your family's friends.
Absolutely. So I'm actually sorry, I have to open something up. It's gonna give me just a second. But we actually do an activity every couple of years in my lab group where we use this exercise. Now it's a little too oversimplified, but it definitely gets to the point of you have to start thinking about these things. So there's these websites where you can put in what you're trying to say, but it only allows you to use the 10,000 most common words. And so with that, it means that you have to try to say what you're, interested in with very little access to vocabulary. So I'm going to drop it in the chat. I think, can I make it where everyone can see it? Yeah. So this is one of the websites. There's a few of them. And is that the one? I'm like, oh yeah, that one, or actually we often use the Upgoer 5.1. I'll drop that one as well. And so we do an exercise where everybody in the lab tries to explain their research using only the 10,000 most common words. I'll share with you, insect isn't in there, bug isn't in there, gut isn't in there, stomach isn't in there. And you can already see from my talk, there's a lot of the things that are around that. And so trying to get creative and using just the most common words, it's a great exercise. It's a maybe too simplistic, but also, spend time with people that are outside of your field and tell them ahead of time, like, hey, look, I want to tell you about my research. Every time you use a word that you don't understand, tell me. And I actually did that once, and I was talking about something and somebody said, what's morphological? And I was like, oh, anatomy. Like, people know anatomy, they don't know morphology, right? And so just these little things that we have to do. And as you're speaking, I'm sure I did it today. I probably was like, when we look at the outside morphology or anatomy, like I try to always correct myself when I use the jargony word. And I actually think sometimes the public likes that. The public's intelligent, they're smart. They might say morphology. I think I know what that, anatomy. Oh, of course I know what that is. And then they keep going, right? And so I think they actually like to be challenged, but you don't want to leave them in the dust with words that, they just don't understand.
All right. Thank you, Corrie, on behalf of all of us here at the Evolutionary Studies Initiative. Warm round of applause, digital and real. And I thank you for your time. We all thank you for your time and spending it with us.